presentation, present to them. And slowly we realized that we are not getting through them because of a problem. And when we looked at the textbook, we realized there are issues with it. There are 11 sentences on RNAi on page 209 and 210 with a figure called 12.2. I'll talk about this in a moment. And this gave us an idea that in order to understand these sentences or the topic of RNAi, we have to change our strategy. So accordingly, my presentation is designed to explain to teachers so that you are equipped to explain the topic to students. I would not be talking of very much hi-fi on this subject. It will be limited to what is required for you to be able to teach this. Those of you who know more than what I'm going to talk, I ask for their indulgence. As Professor Bhatt said, that I have a video which I found very interesting, and teachers have also found it very interesting. You can watch it n number of times, because it talks about RNAi, not only RNAi, some of the basic concepts of molecular biology. So we'll go ahead with the video first. This is about a 14 minutes video, and then I will resume my presentation. Let me now go to the video. I always have a problem of but
मैं सोच रहा था कि मैं उसमें देखूं आपको कैसे दिख रहा है
इस पे कैसे
प्ले करेंगे तो
Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous...
I cannot see, sir.
Every creature has its own DNA.
Not able to see.
Whales.
Sir.
Yes, sir.
People say they are not able to see. I also cannot see. Video.
The video is...
Not visible.
Video is not visible. Okay, I'll just do it again. रुको यार, इसको क्या करें?
चल इसको मिनिमाइज कर देते हैं. इसको एक बार बंद करें आप, जो आपका पीपीटी था.
पीपीटी को बंद कर दूं?
हाँ. आप इस वीडियो के लिंक को ना, अगर हमें दिखाना है इसको.
हाँ.
इसको
छोड़ो.
सही चल रहा है, आपकी आवाज भी ठीक आ रही है.
ओके, ओके.
आपसे वीडियो का लिंक है आपके पास?
रुको, नहीं. स्ट्रेट शेयर अब ये आ गया. So optimize. Now should I make it full?
Flowers, but to go from a chemical
शेयर करना.
शेयर तो किया था. ये रुको. Now is it okay?
हाँ. Play.
Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different for mice, and then for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big. So big that to RNA researchers like Greg Hannon, RNA is more important than DNA.
DNA really works for RNA, and proteins really work for RNA.
Would you get an argument, by the way, from somebody else?
Oh, undoubtedly, sure.
So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and, well, we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code inside the nucleus. DNA, says Greg, never leaves the nucleus.
You ever meet one of those mean librarians, you know, the
Yes.
special reserve section?
The ones that go, pow.
Right. You can take the thing, you can copy it, but you can't take the book because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that's what RNA does. That scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm C that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up, in that sea, you see hundreds of thousands as well. We've made them into little guys with chef hats.
Those would be ribosomes, and in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, all cells. So all these chefs are basically building you. You are made of proteins. And because of RNA, we can copy, we can distribute, and we can cook up you and me. And RNA's been doing this for more than 3 billion years. But there was something spectacular about RNA that nobody knew till just a few years ago, and they learned about it, as we told you, by accident.
Here's a good one, maybe this.
In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower
That looks good.
to attract investors.
So that we could convince venture capitalists, investors, to give us more of the green stuff, more money.
Still, back in 1986, geneticists didn't know how to work that easily with, say, roses. And so
We began with a simple plant, regular garden variety petunias. Petunias being a plant that were easy to introduce genes to in 1986.
And so they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Completely white flowers. We lost pigmentation completely. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake?
A mistake. So we checked everything out, and there were no mistakes that we could find.
So why didn't the petunias turn purple? What happened?
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
And what was going on is, quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks. But some of those recipes, you'll notice, look a little different. And what's in these recipes is not good for the cell.
No, it's decidedly not good for the cell, because the sole purpose of that virus is to make additional copies of itself, and to the point that the entire cell is filled up with this, and the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. So when they see it in that shape,
They say virus.
And
They
they go,
say,
uh-oh.
uh-oh.
and the cop destroys the recipe. And when you say it destroys, is this, should we think like a kung fu kind of thing? Is it like, sort of deal?
Yeah, a little enzymatically, a little thermodynamics. Things like that.
Enzymatically?
Enzymatic kung fu, maybe, yeah.
The cop destroys not only the oddly shaped version whenever he sees that recipe. Oddly shaped, regular shape, that recipe in any form must be destroyed to defeat the virus. And the interesting thing is, until 1998, nobody knew that cells had this defense mechanism.
We had no idea it was there. That's what's so amazing, is this whole mechanism had been sitting there where cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They'd worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The accident was Rich Jorgensen's purple petunia. The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cop saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell.
So there's no possibility anymore of producing the purple pigment because the purple transcripts are gone.
If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes in the cell.
And when scientists realized that every plant and animal cell has RNAi, a way to turn off the recipes, turn off genes, they thought, maybe we can use these cops to work for us.
Okay, Chubber?
Which brings us to Marty Russell, 78 years old. She and her husband used to spend lots of time here at their daughter's nursery.
Thank you, Rosie.
Years ago, she enjoyed doing lots of things.
Her passion was reading.
Oh.
She would read everything.
Golf.
Yeah.
I love to play golf, bridge.
But then Marty began losing her sight.
Couldn't see, and I'd probably get the peppers in with the zucchinis, and there'll be big problems then.
So she went to her doctor, who told her,
You have macular degeneration.
a degenerative disease caused by too many blood vessels growing in the eye, underneath the retina.
As these blood vessels grow, they leak out fluid and blood in the center of her vision, and it's as if you're looking through a very dirty windshield, essentially.
I went home, I was just devastated.
So Marty volunteered to be a candidate for RNAi therapy, something so new she's kind of a pioneer.
She was probably one of the first to get it for any disease whatsoever, and specifically for macular degeneration. Hey, how are you?
Advertiser, how are you?
Good to see you. This is the VIP room.
I feel very honored.
The reason Marty has so many blood vessels growing in her eye, clouding her vision, is there's probably a mistake in her DNA, in a gene that produces too many recipes that say, make more blood vessels. So the chefs cook up proteins for more, and she ends up with too many blood vessels. Her doctor wants her to have fewer blood vessels. But how do you get the chefs to make fewer blood vessels?
It's pretty easy. You want to shut down a gene? Put in a copy of the gene with its mirror image. Signals the cell, better shut this thing down.
We inserted the needle after numbing her up.
So the doctors put literally injected RNA recipes into Marty's eyes that said, make more blood vessels. But they made those recipes look dangerous, like viral recipes, hoping the cop in Marty's cells would leap to it and destroy lots of recipes for more blood vessels, leaving Marty with fewer blood vessels. They wanted the cop to turn off Marty's disease. Did it work?
Marty's vision has improved. It's a very promising result.
I can play bridge now.
Which is very important.
I'm not great, but you know, it's part of my life.
She can see flowers again.
Oh, some of them are just gorgeous.
So apparently they did trick the cop in Marty's cells to reduce vein production, although not completely.
I see the yellow. Inside is just a little cloudy, but I can see it.
There's a lot of questions still that need to be answered. This is not a treatment that is as proven.
Can we deliver recipes to the right cells?
Lovely.
Does the treatment last?
That's beautiful.
All these are big questions. Still, in mice, RNAi has been effective with Huntington's disease, Lou Gehrig's disease, hepatitis, breast cancer. So says Greg, if we ever work this out in humans,
any sort of disease that you can imagine becomes fair game.
all the diseases which would be helped if you shut off a gene.
Cancer, HIV, for example.
Wait, is this because you're just an RNA buff that you're saying? You've just listed cancer and HIV. These are famous, big, fat diseases.
Arthritis.
Well, stop listing them and tell me, is this a prejudice that you're telling me, or is this true? I mean, these are all candidates for this kind of therapy?
Certainly they are.
And finally, we have saved the best for last. The true power of RNAi goes even deeper than finding cures to terrible diseases. Because what RNAi does, remember, the cop's job is to turn off information, turn off genes.
The big problem of understanding, say, the human genome is you have 20,000 genes. How in the world are you supposed to know what each one does? Well, one very good way to start would be to turn off gene number 1 and see what went wrong.
So you could go through all the genes that make up a human, or for that matter, make up a petunia, and turn off each gene one at a time. If you trick the cop to turn off gene number 1, no color. So gene number 1 is involved in color production. Try gene number 2, no petals. Gene number 2 involved with petals, and so on.
You could make too many leaves, they could curl up, they could be upside down. Almost anything could happen.
But getting rid of the gene tells you what the gene does when it's working.
That's right.
The RNAi discovery is just amazing. 10 years ago, when we were sitting around talking about what would we really need to understand the human genome, we all said we would need some magic way that you could turn off any gene at will, just based on knowing its sequence. And what's happened is this discovery by scientists about RNAi has given us exactly that. It turns out that nature already had a way to turn off any gene at will.
And now, with RNAi as their key, scientists will have the means to decode every living thing, to identify the genes that allow us to grow, that allow us to move, that give us beauty and color. RNA is a modest little molecule, but what it gives us is the world.
So that was the video. Now let me go back to my presentation. Okay, so that was the presentation. And now let me go back to what I said that I will talk to you about. The text, as I said, 11 sentences are given about RNAi. And if you look at, it says pest-resistant plants, and then suddenly they say a nematode, Melidogyne incognita, infects the roots of tobacco plants and causes a great deal of loss in yield. And you have seen this. There are some keywords like complementary dsRNA. I have problem in understanding this, but still, we will talk about what is dsRNA. Then it says using agrobacterium vector, nematode-specific genes were introduced into the host plant. Figure 12.2. Figure 12.2 is nowhere close to what is described here. Then figure 12.2 is again mentioned. It says the transgenic plant therefore got itself protected from the parasite. And the figure, if you see, this is the figure. So you have two A and B, and the legend also does not tell you what these are. So based on that, I thought we will try to explain each of these sentences, 11 sentences to you, so that the whole process of RNAi makes sense to you. From the video, you would have got the information that RNAi is a process by which in nature genes are turned off. So by understanding the mechanism, we have been able to turn off genes which we want to do in plants, animals at will. And how do we do that? That's what my talk is all about. Now, just to recapitulate the central dogma, you already know that the information flows from DNA to RNA and finally to protein, which ultimately results in the phenotype or trait. Yesterday, Mithali talked to you about how do you sequence DNA and what all information you get. One of the methods by which you can identify function of a gene, as explained in the video, was RNAi. Using RNAi, you know the sequence, so you create a RNAi for that sequence and look at the plant or animal or the system and see what process has been affected. So you identify the function of a particular sequence, link it to a process. I hope that's clear. Now, initially, there was a strategy which was used, which was called Antisense Rna Strategy. So what would do is that if you have a sense strand of mRNA, if you introduce a construct into the same cell which will transcribe an antisense. So what would happen is this antisense, because of its base complementarity of the mRNA, will bind to the mRNA and will degrade the mRNA so that no peptide or protein is produced. So translation was inhibited. So this technology was called antisense technology. And so if you have a gene, if you have a complementary copy which is being synthesized as an RNA, the messenger RNA would combine with antisense to give you a duplex formation. And it was observed that the translation was blocked and the character which you are trying to inhibit was inhibited. And the proof of this was the famous example, which I think all of you would be quoting in your classes, the flavor saver tomato, which was the first transgenic plant which was commercially allowed to be grown and sold, had used antisense of a gene polygalacturonase, which prevented the ripening of the fruit. I think you're familiar. So that's one. So if you have antisense, the translation is blocked. That was known in 1990. In the video, you would have seen that Jorgensen's experiment told you that if you introduce the sense of, that is, another copy of the same gene, for example, in this case, a chalcone synthase, which is related to the color production in plants, what he got was a series of flowers with varied variegations, and one which was totally white, the color was lost. That means the endogenous gene which was present in the plant was also blocked, in spite of the fact that the workers did not introduce an antisense, but used a sense strand, which would have made more of this mRNA but unexpectedly, this caused the blocking of the pathway and resulting in loss of color. So this has resulted in identification of why should this happen, and then people from different fields, from Drosophila, from people working in nematode, got interested, demonstrated that this indeed happens, that if you introduce a sense strand, a double-stranded DNA, a double-stranded dsRNA, double-stranded RNA is formed, which is the cause of the loss of function. And this process, as it was known as RNA interference, RNAi, and in the process, there were some molecules which were discovered, which we'll talk about. And for this work, Craig Mello and Andrew Fire got Nobel Prize. Being a plant scientist, we felt bad because there was significant contributions which had come from plant science. For example, Sir Balcom from UK and Jorgensen's initial experiment had, in fact, identified the process which was not known earlier. Even Fire admitted while receiving the Nobel Prize, he said, "I feel slightly guilty to be here, given the strides taken by others in the field." Be it may, as it may, but the process was discovered in plants, and it was further refined, and the mechanism was worked out in C. elegans, a nematode, a free living nematode, and also in Drosophila. The terms which I'd be using is RNAi. We have already said this refers to RNA interference. That is, you are interfering with the message which is produced so that further translation of this message into protein does not take place. So the whole process is known as RNAi. In this process, we will also talk about dsRNA, which I have already talked, that is double-stranded RNA. Then shRNA, which is, I will explain these. shRNA refers to short hairpin RNA, and an important molecule called siRNA, which is small interfering RNA, which is double-stranded with 21 to 23 nucleotide long. Another term which I'll be using is called RNA-induced silencing complex, or RISC, which in the video, it was called the COP, which would destroy the mRNA, endogenous mRNA for a message. So this complex is called RISC, RNA-induced silencing complex. I think if you know these words, I think we can go ahead. Now, what happens is that if you have in the system double-stranded RNA, that is a copy of sense and antisense together, as I said, they will form a double strand in the system. I'll talk about what is this hairpin. So you can introduce the so-called hairpin double-stranded RNA or shRNA into a system, into a cell. I will tell you how. This shRNA, if it is introduced into cell artificially, there are systems which cleave this, get it associated with RISC, which is RNA-induced silencing complex. So the siRNA, the antisense strand, forms a complex with the messenger RNA and degrades, it slices the messenger RNA, and the information is lost. So this is very brief, in simple terms, the process which takes place. Now, there are two things. Your textbook says dsRNA. How do you get this dsRNA? And what is this hairpin RNA, which we'll talk about. So so thus RNAi could be used to knock down a gene function. That's clear to you. Now, RNA interference induced in insects and nematodes after injection of plant-expressed hairpin RNA offers promise for managing devastating crops and pests. Large number of plants, crops are infested with insects or nematodes. So if we can use this technology of RNAi, which will kill an important message required for the development of the nematode or the insect or its interaction with plant, we can reduce the infestation. That's the idea. Now, let me now shift gear and talk to you about something which is required to understand this. Now there are several nematodes which cause severe yield losses in several crops. And one of the important one is called Melidogyne incognita, or it is called root knot nematode. And you would have seen in the textbook, the photograph would have shown you with roots, with galls or nodules kind of things, knots, which are present on the root. So what happens is those galls contains these nematodes which suck away the nutrient of the plant, and the plant growth is affected, thereby causing yield losses. There are other series of other nematodes which cause different ones. We would just concentrate on this Melidogyne, which is mentioned in your textbook. Okay, so this is a field view of a place where nematode infestation has taken place. You can see the plants have stopped growing or have a stunted growth because these nematodes are soil borne. The other important feature of this nematode is that it's an obligate parasite. That is, it will live in the soil, but it will grow and multiply only when a suitable host or a plant will be available. So it divides and multiplies only when a suitable host is available. So if the soil is infested with the nematode, the plants in that area would not grow, and slowly this spreads. The nematode spreads. Next year the area would grow, more and more nematode would be present in the soil. Since they are buried deep in the soil, you cannot do with anything with them. So you can't spray an insecticide or nematicide because they are not at the surface. So they hibernate deep inside the soil. So getting rid of these nematodes has indeed been a problem for agricultural scientists, farmers, and so on and so forth. So this is the life cycle of this plant nematode Melidogyne incognita. As I said, it's a parasite, and the female is the one which produces the egg mass. And these egg masses are released from the roots into the soil, and this stays buried in the soil. These eggs are quite hardy, so they stay in the soil for several years under harsh environment. Only when a suitable host is found that these eggs hatch, they provide what are called as juvenile, and this juvenile one, which is called J2, is the one which infects the roots. So it enters the plant root, penetrates, goes and forms a gall, and there it undergoes, plant also undergoes differentiation, nematode also undergoes various metamorphosis, finally as a female, and this female grows into bigger size, lays egg, and this cycle continues. This is what causes the disease or pathogenesis. Okay, so can we use RNAi to knock this, some of the functions of this nematode? That was the question. In C. elegans, which is free living, what you do is if you soak it in a solution of RNAi or dsRNA, a function is inhibited. That is what Craig Mello did. Or you can feed bacteria which contains this dsRNA, E. coli. The C. elegans feeds on lives on bacterial colonies. So you can introduce into C. elegans these dsRNA through soaking or through microinjection or feeding bacteria that contain these dsRNA. But how do you deliver it to a plant parasite, which is obligate parasite? So what was thought was that if we can introduce a dsRNA construct against nematode genes into plants, ask the plant to make these dsRNA, and once the nematode infects this plant, it will be ingesting this RNA and can be now interfere with the development of this nematode. So now, how do you make the plant produce dsRNA specific to the pathogenic nematode? That is the question. Now, for doing that, I think Dr. Monica and later I think Deepak also talked to you about vectors. So there are vectors. So you can have a vector where the targeting sequence, which would be of the nematode gene, 300 base pairs, and a antisense copy of that also in the same. So you have the same sequence with in between there is a spacer, and then you can have the antisense in the same construct. So what would happen is the promoter would transcribe this RNA as such. So you will have sense, antisense with an intervening sequence or a spacer in between as a product of RNA. Now, once this product is generated, what will happen is it will form a loop. This is the space, this is the spacer which does not have complementarity, but those sections which are sense and antisense will form double-stranded. So this is what I was referring to as the hairpin loop, short RNA hairpin loop. So once this molecule is produced by the host genome, it will be acted upon by dicer and the COP or the RISC complex to give you siRNA, which will be produced in the plants. So the idea was that using agrobacterium or a gene gun, you can introduce a construct which will produce this dsRNA into plant, and then the plant will be able to make this shRNA, which will be converted to siRNA, which will inhibit the, mind you, the nematode. So if the nematode now infects this cell, it will find these siRNAs which are ready to cleave the target mRNA. So this target cell is, so there is a problem with this cartoon. Here the host genome is plant, but the target cell is the one which is of a nematode. So this would be ingested, this target mRNA. So I have to modify this. So this target mRNA will produce this, and these are available for the nematode, which will then target the mRNA which is produced by the nematode. The photograph which is produced in your textbook has come from a paper which was published by Dr. Subramaniam from IIT Kanpur, which it's a short communication. This was the first report, which says host-generated double-stranded RNA induces RNAi in plant parasitic nematode. And this is the photograph. There are also inserts. You can see these inserts here, which are not explained in your textbook. So this is the A frame. You can see this is the plant root, and you can see these knots-like or galls-like structure. These are the, and with an arrow. So these are the parts which have been, now a nematode is growing, the cell is multiplying, female is producing egg mass, and so on and so forth. So this is infected plant root, and insect shows a egg female which is laying egg, it is releasing the egg mass. This is a controlled plant where infestation has not taken place, or this is what it would look like. But if you ask now this plant to make RNAi, siRNA against a nematode gene, you can see that the gall formation has significantly reduced, in spite of the fact that in some, you can see some nematode in the insect, they show their nematodes are present, but they are not able to grow and multiply. So I hope I have provided explanation for this figure. Okay, the other thing which I would just add on to, we talked about selectable and scorable markers. So we also got involved into this. Briefly, I will talk about it and raise a question about it. So we had, for example, we had a marker which can be visualized by color. So it is called beta glucuronidase. You will see a blue color wherever the gene is expressed. So look at this. This is control plant, root of control plant. You don't see any color. This is root of a control plant where you see color when nematodes are infected. So that means, and it is present in all the roots. So all the root systems somehow realize that it is now being affected or it's being infected by nematodes. You can see nematodes. Another example of wherever the nematode is present, in this case, in contrast to in all roots, where what you find is if a nematode is present, which you see, which has been stained with purple color, you can see the plants have started expressing a gene which gives you blue color. So this was in order to identify nematode responsive promoters in plants. Now, the question which I'm leaving you with, what would be these promoters of use? What use you can put them to? So the two questions which I'm leaving you. What would be some of the desirable characters in a gene which you would select for RNAi for creating nematode resistant plant? First, what is the utility of such promoters which I have discussed with you? So with these two questions, I have a link for you. Just type ISAAA, and this is a site where you have Pocket K, small pocket books for various aspects of biotechnology. So the 34 number, Pocket K 34, talks about RNAi for crop improvement. The question is, has this RNAi been put to some use? What are the examples? I'll just show you a table from this pocket in a moment. There are other very good animations and talks available on YouTube and others. Just type RNAi and Nature. The Nature journal has produced a wonderful animation of the whole process of RNAi in a eukaryotic cell. Now, let me just go back and present to you the, give me a moment, and I will share the Pocket A, the table which is present in Pocket A. Which gives you examples of various uses this has been put to. Deepak, इसको minimize कर दो. This I have to share.
शेयर हमारे से डायरेक्टली सेकंड शेयर कर सकते हैं. इसमें शेयर स्क्रीन करें. शेयर स्क्रीन उसमें आ जाएगा ऑप्शन जो आपको शेयर करना है. देन अगेन उसको पावर पॉइंट कर सकते हैं. शेयर.
Yeah, so this is the table which I have shared with you from the K book, and you can see the properties which are just a moment, view single page. Okay. Yeah, so you can see enhanced nutrient content in tomato, in wheat, sweet potato. So these are RNAi technologies which have been used to create these. Reduced alkaloid production, let us say in opium, in tobacco, heavy metal accumulation, reduced polyphenol production, and so on and so forth. You can think of ethylene sensitivity, that is for early ripening, reduced allergenicity in peanuts, in ryegrass, and reduced production of, let us say, tear in onion. So if you can, so these technologies have been used. There are n number of references. If you are keen, you can go back to them. And with this, I think I have come to the end of my
again in that Zoom app. Share
screen. And I hope I have, and if you have any questions, you can always shoot those at my email or to Genetics Congress Trust. And thanks a lot for your patience.
Okay, sir. You can stop this slide share. Hello. Unmute yourself, sir.
I am audible?
Yeah, now you are audible.
Earlier, when I was presenting the slide, I hope I was audible. Okay.
Yeah, you are audible. No problem. You close this share slides at the moment.
Okay. I'll do that. I'll just give him a moment. I will do that. Yeah.
Okay. Now I'll flag the questions that we have received. Why it is called antisense, although it makes sense. So you better clarify sense, antisense. Hello?
Okay. There is a convention. So do you know DNA is double-stranded? If you look at the mRNA sequence, I'm not audible. Am I audible, sir?
There is some
What's
breakage.
up?
I do not know. Yeah, is it at your end or at my end, or everybody's? I do not know.
Yeah,
Sir is audible. Sir.
you are audible.
Yeah.
Because of presence of this. I don't know. Is it?
Okay.
I have unmuted myself. Yeah. Is it
Yeah.
okay, sir?
Yeah,
Okay.
yeah, I can.
So the answer to that question is DNA is double-stranded, and one strand of DNA is converted into mRNA, which is a complementary copy of that DNA. That mRNA carries the code for protein synthesis. So it carries those codons which are read by tRNA and ribosome to convert them into another language of proteins. So that mRNA which makes sense is, and from the sequence which is carried by DNA, remember, one of the DNA strand is copied. So one acts as a template and a complementary copy of that goes as mRNA, which makes the sense. So this sense, the strand which makes sense as far as coding is concerned is called sense strand, and the other complementary copy which is used as a template for copying is called antisense. I hope I have made this clear.
Okay.
So in the cell, when mRNA is being synthesized, normally DNA is being copied and mRNA is being sensed. That is a sense strand. Now, if you can an RNA which is complementary to the sense, that is, you produce mRNA which carries exact same sequence as antisense strand, so that will be an antisense mRNA. And both are present in the same cell. Because of the base complementarity, these two mRNAs would come together and form a double-stranded structure.
Hello.
Yes, sir.
Sir, there are a number of questions. You need to tell how the gene to be silenced, let us say in nematode. Hello? Are you able to hear? Hello?
Yeah. Yes, sir.
Okay. How do you identify the gene to be silenced in the nematode? How do you take that? How do you transfer it to the plant? And how do you actually construct sense and antisense? I think
I have to go back to my slides.
You will have to go to slide, but you can use the restriction, blunt end restriction.
I'll just. I have two slides.
Yeah.
Give me a moment. Let me go back to my slide presentation and.
How do you choose a gene? Where do you isolate that particular DNA sequence? How do you construct double-stranded? And how do you introduce? I'll come to the next part later.
Okay, just a minute. So. Okay.
Share.
Share. Share. Is this visible, sir?
We are not able to. I am not able to
But
see.
sir,
Manji.
you are able to see now?
Yeah. Yeah.
Okay. So look at the pointer. So here
No,
you have.
first talk about the gene from where you.
Sir, I'll come to that. First, let me explain this. I'll come to the gene. How do you get the gene? Just because this slide is open now. So I'll come to how do you get this. What is this? Okay, so here is the target sequence. I'll come to how do you get this target sequence. Okay, so this sequence and an antisense of this is introduced into a construct. So there are vectors. Yes, few days back, Dr. Monica talked to you about PBR322 or PUC and so on and so forth, which have multiple cloning sites and so on and so forth. So we have vectors which would create these kind of sequences once you have identified the target sequence. So what will happen is, so you have this target sequence here, an antisense of this, and if this mRNA is produced, what will form is these two will come together to form double helix. The intervening sequence will form a loop. Now, let me go to another slide and also talk to you about, for example, this is a vector which is called P. Hannibal. So you see that you have several multiple cloning sites, and it is synthesized, it is made in such a way that you can create a target sequence and inverted sequence of the target, and this vector can be now introduced to transfer this gene, the so-called double-stranded RNA, which will produce dsRNA. So having said this, if that is clear, let me come back to the question which Dr. Bhatt asked. How do you identify the gene? Okay, how do you clone the gene? Now, this is the question which I, in fact, posed to you, that what do you think should be the characteristics of a gene which is selected for RNAi? So these genes would be such, I'm giving you one of the characteristics, that they interfere with the development of RNA of the nematode. Now, how to identify these genes? I think yesterday Mitali gave an excellent talk of genome sequencing. So most of these nematodes have also been sequenced. Based on the sequence data, you can identify what are the genes which are required, which are essential for the or are essential for interaction with the plant and so on and so forth, for their infectivity. So you can identify the sequence, and Deepak talked to you about PCR. You can clone these fragments using specific primers directed towards these genes, and you have these double-stranded DNA which you can amplify or clone it in bacteria, which are available to you for cloning. You can use suitable restriction sites if they are present, or also you can create restriction sites. Dr. But have I answered the question? Was that the question?
Yeah, basically they wanted to know how it is constructed.
Right. So that is what these vectors are available. Yes. As I told you,
they might be available even if it is not available. Yeah. If you have a single enzyme cut, let us say in 3, and insert could be in 2 orientations.
Both the orientations.
Making use of that also.
Have both these orientations.
Yeah. Maybe I think we will explain it on Saturday with additional.
So I need. So we have to talk about promoter, and so we have to explain. I think essentially that in any system, if you have a proper signaling system with a proper promoter and terminator, whatever RNA you introduce, DNA you introduce, it will be transcribed. It should make a sense. Any piece of RNA which is properly placed with a promoter, let us say a T7 RNA polymerase promoter or a LAG-Z promoter, will be transcribed. So you will have an RNA produced. So then we will have to talk of expression vectors. But essentially what you are trying to do is put this RNA DNA construct in plants with proper signal, which will be recognized by a plant. In the example which is in your textbook, a CAMV35S promoter, which is a viral DNA virus promoter, has been used to transcribe the construct which has been introduced. So whatever is present between the promoter and the terminal sequence will be transcribed by this plant cell. Copy, because they have a cell ability to form double stranded structure, the complement, because of complementarity, those will come together and form a hairpin RNA. And then the system, which already has dicer and the COP, will take care. Yes, sir.
Sir, still I'm finding more questions. How introduction of one DNA sequence produces two strands of RNA? That is the same question, falls back to the same, that they are unable to understand that the stretch of DNA
is converted into RNA.
Reversed in one. No. See, basically, how introduction of DNA produces two strands of RNA, sense and antisense.
Let us leave it.
Your basic
let me make a slide. Isko abhi chhod dijiye. We'll make a slide with different colors.
I think on Saturday
we can
we'll address this again. Because that needs to be clarified in a much better way, because the conventional textbook we are used to, an mRNA comes from the now couple of more questions. Why one RNA strand is required for ultimately in RNAi, but we start with dsRNA, double stranded RNA?
No, so the antisense strand is in fact used for binding to the mRNA which has to be degraded. And that is, if you don't have a complementary copy, so siRNA essentially is that you have two strands. One which is the copy of mRNA, the other is the mRNA itself. So that strand in fact binds to the end of the RNA, and the complex takes it and then degrades. So you get a double-stranded RNA again with mRNA because of the complementarity. And these fragments, small fragments, wherever they go and bind, will be degraded by the COP or the RIS.
What I feel is you need to tell that dicer can only cut double-stranded RNA, not single-stranded. That is why we need the first phase, a double-stranded RNA.
But the question was about, okay, chaliye. Yeah. Let me, if you look at this, it says that dicer removes the intervening sequence from a hairpin to produce a siRNA duplex. And then the whole RISC binds to it. And you would notice that RISC here, the one which is shown in black, is complementary to the RNA. So siRNA forms a complex with mRNA, and the mRNA is degraded in this cartoon.
Okay. One other recurring questions in the past also. We produce dsRNA in the host. It is consumed by the insect or the nematode. And why not this RNA not degraded in the guts of insects or nematodes? How is it able to silence in the ultimate organism, be it nematode or insect? How it survives in the gut and how it is able to reach to the mRNA present in the host, in the pest. Sorry.
Okay. It is also soaked from the surface of the nematodes. So it differs from system to system.
So for example, insect also it is used where it is,
Insects
consumed.
also it is used. Yes. So these the hairpin RNA, which you would have seen, it's anything which is double stranded is stable. So unless it goes inside the cell where dicer is present, it will make it single stranded. So compared to single strand RNA, double strand RNA is more stable. The other is the number which you feed is large. Even some of them, if they get degraded, but even if few can reach the point, you would notice that once they are present, they kind of amplify themselves. They produce more and more. The other question which some of you might have, or I don't know whether it has occurred to you, that is it transmitted from generation to generation? Again, there is a lot of biochemistry which we understand about how these RNAi silencing things are transmitted from generation to generation. In some cases it is not, in some cases it is, depending on whether you have a specific RNA dependent RNA polymerase and so on and so forth. But I didn't want to go into details. Those of you who are interested, there are very good talks on these available on YouTube. And I just wanted to leave it as here, which is good enough for your students so that you can explain it to students. For those who want to know more, or as I said, some of you already know more than what I talked about. There are n number of things here. In some cases it is not, in some cases it is, and so on. So it varies from system to system. In some cases you can feed it, in some cases you cannot. For example, so the idea was that if you, for those obligate parasites in case of nematodes, how do you introduce them into them? So that's what I wanted to talk about. But it varies from system to system.
Yeah. In fact, one
deep mechanism.
Hello. Hello.
Yeah, go ahead. Yes, sir. Yes, sir. Go ahead.
Deepti Avasti has asked an interesting question.
Go ahead.
If mRNA in the sense and antisense are introduced separately by two different vectors, will it produce double-stranded RNA? Which, yes, it will be. You can explain.
So
another way of doing it.
Right. So you remember I did talk to you about the first example which I gave you is antisense technology. So you already have a sense mRNA which is being produced by plant. You have another DNA construct which will transcribe the antisense strand. So the cell will have a complementary copy of mRNA which will combine. Now, remember DNA is double strand. Again, the same point which we have been referring to. But which strand will be transcribed will be determined by the promoter, okay? The orientation of that. So the promoter, DNA is double strand. So which strand will be transcribed will be determined by the promoter. So that's what I said. Any strand of DNA, if you have introduced, which will be double-stranded into an organism or a cell, will be transcribed depending on where promoters are present. So let me give you an example. There is a vector available which where if you can clone the DNA, you can transcribe both these strands. That means RNA produced, and once you have 87 RNA polymerase, the other side it is sp6 RNA polymerase. So if you want to produce RNA from one of the specific strands, you can use that system in vitro. So in plants, in animals or anywhere else, what you can decide which strand will be transcribed. In case of when we say dsRNA construct, what we are doing is we are using a single promoter, and the DNA is such which carries sequences in between which are complementary to one another. So what happens is DNA is double stranded, so it will have another strand which will keep it double stranded. But once the strand which will be transcribed carries these complementary strand together, the RNA would come, those complementary sequences would bring the RNA together, forming a hairpin loop. That is what happens. So there is only one strand which is being transcribed, but based on our information about the gene, what is sense, what is antisense, now you have a double-stranded RNA with a sense strand and antisense strand coming together.
Sir, there is another question. Because in the NCERT text, things are given here and there. They are not connected. They say that in biotech experiments, we use techniques like gene gun and vectors to deliver desired gene. So they again say, how can you introduce dsRNA into cell, into plant cell? It is not dsRNA.
It is a DNA construct which you introduce, which will produce dsRNA in the cell. So as I said, any piece of DNA which has a strand which carries complementary sequences, and if it is transcribed in the cell, it will produce a dsRNA. And this can be introduced like any other DNA which you do if you have studied Bt toxin gene or any other gene. So it is introduced using agrobacterium or gene gun, or there are other methods which I think you are familiar with. But if you want, we can talk about it separately. What are the methods which are used and how do you construct and introduce these genes into plants? So suffice to say that what we are doing is introducing a DNA sequence which will result in production of dsRNA in the cell once this strand is transcribed by the promoter.
Yeah. There are two questions which are related. You said that RNAi is present in humans. If it is present, what are its effects? What is its function? The second one is, can we use RNAi for COVID?
Yeah, the answer to these questions are yes and no. If you listen to the video once again, he reveals out a number of diseases like arthritis, HIV, you name it. COVID is one viral disease. Yes, theoretically, yes. But how effective these are, you would notice that in these cases, I have used a term called knockdown, that RNAi knocks the expression of the gene down. It is not completely knockout. Please remember this. So in some cases, I would prefer that the RNA or the target gene is completely knocked off. So there are now recent times methods which are more powerful, better. You would have heard of CRISPR-Cas. So RNAi is kind of going into the background now because the modern tools allow you to completely knock out a gene function. In this case, as you would have noticed even in the example which I have showed you, that you have flowers with different variations. So in each of these individuals, the knockdown effect has been different because you are unable to control how much of introduce RNA, all the mRNA would be completely knocked off. It will be knocked down. So it depends on several systems. So yes, theoretically it can be. And as I said, this is present as a defense mechanism against specific viruses since time immemorial in all our mammalian system and plant systems. So probably it is against viruses which have specific sequences, that is, if they form somewhere a double-stranded structure, it will be knocked off. So in that sense, it is limited. So if you can, if you have a virus which have those kind of, which will form those double-stranded RNA structure, siRNA will be activated very fast. If it does not, the RNA would not be active. The siRNA system would not be active.
Sir.
Yes.
In the NCRT book, they say that one source of complementary RNA is infection by RNA virus. How RNA virus infection gives rise to dsRNA?
Repeat it again, sir. The source of complementary strand is RNA.
No. In the book, it is said that RNA infection by RNA virus, infection by RNA virus is one of the sources of complementary RNA. If so, how are the dsRNAs produced?
Complementary RNA to what?
No. One source of complementary RNA, complementary to the viral RNA, perhaps. This is what is said in the book. One source of complementary RNA is infection by RNA virus. That's what is given in the textbook. I do not know where exactly.
Okay, I have to look into it. So we have to, maybe Shally is talking today or tomorrow?
Tomorrow.
Maybe we can ask this because RNA, each one has a different replication mechanism. So in some cases you can have a sense strand infection. In some cases it has to be converted into the complementary copy. Is that they are talking of?
Yes, that's what exactly happens. If you have a negative strand and then it is converted into
positive strand.
Let's say from cDNA again they produce the mRNA, and then if the viral original RNA is there, then it can make a double-stranded RNA.
Okay. So
I'll ask Dr. Shally to
I will ask Dr. Shally to explain that. I at this moment I don't know the answer. Yes.
Yeah.
And the sentence has to be reread again. Yeah. Okay. Go ahead.
Yeah, we will have to look at this one. So more or less all the questions, in the sense overlapping questions, some people want you to explain the dicer, how it exactly does it, how it cuts, and RISC complex. Can you go back and then try to show that?
The same slide, or they want something more? I didn't want to go into details of this because then you have to talk of Argonaut and the whole complex. Yes. So I want to keep it
possible without
so maybe we can have a separate session for those who are interested in just RNAi mechanism specifically.
Yeah. That's more or less complete, because all of the people are asking that how it reaches ultimately the cells of the target organism, be it insect or this and that. It is still far more complex.
Yeah, the exact mechanism we do not know. So, for example, when the C. elegans takes it up, it initially obviously goes into some of these cells, and then slowly it goes to all the cells, even in these insects. So this is ingested by the nematode, which feeds on the plant. It feeds on the plant. So dsRNA goes inside, and in the cells it is absorbed. And how exactly the mechanism it's different, as I said, in different detectors different. How it spreads, but the at one place, several nematodes will enter. So if one nematode enters and it feeds, and dsRNA spreads and has its effect, it's unable to multiply and divide it at the J2 phase. Nematode enters ends with the same fate. That's what we think happens. Some of them still, the infection control is not 100%. You still have about 30% of the galls in nematode ending the gall formation. So it's not a uniform effect, but yes, there is a higher percentage of this effect seen. As I said, this is again, as I said, it's a knockdown. So it has its effect depending on how much of RNA is present, what is the gene which you have targeted, and so on. So
one other thing, maybe you can tell them that RNAi is an amplifying signal. In the sense, if some cells are, starting RNAi expression, there will be more and more. Can you hear?
Which you get once the dicer cuts it into several pieces. So more and more such siRNA. So it's a self-amplifying. I think I did mention, but if I have not, please. So it amplifies. So it signal can amplify.
And signal can move from cell to cell also.
That is the last mRNA fragment.
Your audio is breaking. Your audio is breaking.
Okay, I am video बंद कर.
Video बंद करके कर लीजिए.
एक मिनट. Where is that? Mute my audio. Stop video. Okay. Is it okay now?
Yeah, now it's okay.
Okay. So as I said, a number of the mechanism of how it is ingested but as Dr. Bhatt points out, that this is an amplification process. So what you get is, you get several of these siRNA made, number of them. So as you go ahead, the number goes more and more. And they have varied effects. We only talked of its effect on the mRNA, but it has effects before translation also. I'm not going into those which are at the level of transcription. RNA has an effect and so on and so forth. But suffice to say that there is a knockdown effect of these siRNA, and the mechanism varies from system to system. That is why each system, it has to be evaluated case by case basis, gene based on what is the gene, what is the target gene, and what is the system.
You can maybe mention that there are miRNA genes in every individual.
Okay, yeah, I can. So it's similar to siRNA. There are genes, small RNA genes, which form these double-stranded structures, which are called miRNA, which are called microRNA. So means all these organisms have been found to have genes for these miRNA. And these miRNA have been found to regulate gene expression. Yesterday, Mitali was talking to you about genome sequencing, and she talked about only the coding part. But there are genes within the coding part, outside the coding part, which will code for a miRNA, and the sequence of these miRNA then go and bind to specific target RNA to inhibit them. So there are target genes for them. So this is another unfolding chapter in the regulation of gene expression in eukaryote, where a similar structure of, it's not only virus, but another structure called miRNA, which is microRNA, has also been found. Large number of microRNA in different plants, animals have been identified, and they have been identified with a particular process, let us say in plants, identified with flowering, with development, with hormone response, with diseases, and so on in humans also, with diseases, with stress, and so on and so forth. I didn't want to add that here because that will add to complications. So, but yeah, but as long as you are aware that a similar system exists for gene regulation, not only virus control, but also normal gene regulation. A large number of miRNAs are involved in gene expression. Regulation of gene expression.
Yes. Basically, that's what. Unlike complete knockout of a gene, in many cases, we have to modulate the expression levels, for which RNAi or microRNAs are very useful. The level can be altered. It is not completely knockout, it is knocked down, as Dr. Srinivasan said.
And these are called miRNA, that is siRNA, and these are miRNA, or these are called microRNA. Yesterday you were talking about microsatellite. These are microRNAs. Nothing to do with microsatellite, by the way. Yeah.
Yes, it's only because of the size they are micro, called micro.
Yeah.
I think with these, we have taken all the questions. There are one or two which I'm reserving it for Saturday, because I feel that that requires some explanation of a fresh slide. That's why I have not flagged it here. I think with this, I thank Dr. Srinivasan for an excellent presentation, and all the participants for joining us in this morning session. We will be again joining at 2:30, and please make sure that you use the afternoon link to join the evening session. Thank you very much. Thank you, sir.
Thank you very much, sir. Thanks to all.
Thank you, sir. I'll close this meeting now.
Okay.